So yeah, we'll talk about, so the title is Cortex-wide topography of 1/f exponent in Parkinson disease. So I guess the only words some of you can understand is like Parkinson disease and maybe cortex. And then yeah, I will explain you what all this mean. I hope it will be clearer in us. So I will first talk a bit about Parkinson and why we are interested in the whole cortex. So the cortex is mainly the gray matter in the brain. So where we suppose like all function is main function happening. Yeah, my main cognitive function. Sorry, and then I will talk a bit more on how we can study Parkinson using some brain imaging technique, and I will finally explain you what we did, like how we deal with this kind of data and what we call aperiodic activity and 1/f exponents, and then I will end with our main results. Okay, so Parkinson disease is mainly it's mainly known for a loss of what we call dopaminergic neurons, which are situated in the center of the brain, which is this black area here. And so dopamine is like a neuromodulator that helps in many tasks like learning, motor tasks, or this kind of things. So it's very important, and so we know a lot about what's happening around this area because these neurons project, this dopamine mainly project to this area, and this is an example to show you what we know. So we know a lot about what changes in firing rates, for instance, synchronization, connection, and oscillation in this brain region. So all these boxes are like some nuclei that are present close to this region. So this is just to show you what we know, but we know much less on what's happening during Parkinson disease in the cortex. So that's what we will try to explore, and I will try to show you why it makes sense to look at the cortex. So first, we know a lot about motor symptoms in Parkinson disease, but in fact, there are many other symptoms. So this is just an iceberg representation to show that there are many other problems in Parkinson, like sleep difficulties, loss of olfaction, also some of them which are very important. The second reason is that this is why we would like to study the whole cortex, because these are mainly cognitive things, and you have also problem of cognition. And the second reason is that dopaminergic neurons the dopamine also project to other parts of the brain, not only the one close to the area that I just told you about, which is called basal ganglia. And it also projects to the cortex to some extent. And beyond dopamine, also there are other neuromodulators, neuromodulators are basically some chemicals that regulate and help the functioning of the brain. So there are other ones than dopamine that are altered in Parkinson disease, and they are somehow involved in maintaining also what we call excitation and inhibition balance, which is maybe you don't know, but in the brain there are like excitatory neurons and inhibitory neurons. And there is, when you look at the dynamics of the neural activity, you always see that you need a balance between these two, so to avoid things like epilepsy, for instance. So then when we think about this excitation inhibition balance, we can ask the question, how do we measure this? And it's in fact very hard to measure. So having the assumption that this EI balance is perturbed in Parkinson's disease, and mainly in the, we expect it to be perturbed in the cortex. We looked for a way to non-invasively measure this in PD patients and also in LC control. So now this is the main question, and I will do a bit, go a bit more into the method part, and then I will come back to this question. So to measure brain activities, there are many ways. So these are the main ways that you can use in human. So what you see here is like EEG, which are mainly measuring the electrical field, which is produced by the neurons when they communicate, so they basically send them ions. So this creates the move of ions to create an electrical field, and we can measure it. And images like the same thing. So you create the electrical current will create a magnetic field, and we can measure it. And this is, as you can see, it's a much bigger device. And then there are other techniques which are like more based on magnetic resonance. So there is fMRI and diffusion MRI, so fMRI gives you an activity of the brain, but in an implicit way, because it measures like blood oxygenation, and diffusion MRI gives you some insight on the connectome, so on how the neurons are connected, but mainly through the white matter, that's the brain matter, so not the cortex. And the last ones are more invasive ones. So LFP is basically an electrode that you put in the brain, and ECOGs are like some electrodes that you put at the surface of the brain. Yeah, I forgot to tell you, don't hesitate to ask me any question during the talk. There will be a sampling rate of your measurement. So it depends a lot. fMRI is very, is it sampling in the sense, the temporal sampling? If it's this, you can open your mic, I guess, if you want to ask it. If I'm not going in the good direction, but
yes.
Yeah. So if it's time, it's like images like milliseconds. You can have a millisecond resolution, even a bit lower, and fMRI is more on the order of the second, and LFP is also like on the millisecond. Even LFP are very very good time resolution. Even though I'm not an expert, but I think it's like some kilohertz. And then there are other way of measuring the brain. You can use proteins, and that you can measure also through the cerebrospinal fluid. But what I will focus on is mainly images, so I will go a bit more into details on this method. So as I just said, this has a very good temporal resolution, about the millisecond, which is kind of good because the neural activity, we know that the spikes, so the electric pulses that the neurons use to communicate, are of this order of magnitude, so that's very good. They have a spatial resolution which is not so bad compared to MRI. Sorry, it's still not so good, but it's better than EEG, for instance. I will explain you why. And why it's just the next point. Like, it does not depend on head geometry, because basically the magnetic field cross the brain without being disturbed. Not so much disturbed, because EEG, the electrical field, is very disturbed by the different conductances that you have in the brain. So it's hard to do what we call the source reconstruction. I will explain you what it is. So the drawbacks, the main ones are it's expensive, so there is not, that explains also why there's not a lot of them. So there are, I think, around 200 on the whole planet, so not so much compared to EEG that are almost everywhere. You lose some information because you cannot have a magnetic field which is in some direction, so you have only one direction. It's very sensitive to noise because the magnetic field that you measure is very, very, very small. So yeah, I gave you an example here of the order of, I think it's called femtotesla, so 10 to the power minus 15, and you measure at the same time the magnetic field of the Earth, which is 10 to the power minus 5 Tesla. So the squid are used to measure, to be able to measure this so low field. And another bad thing compared to EEG, which so EEG decrease the signal decrease as the square 1/square the distance, and in the case of magnetic field is 1/the distance. And so yeah. Then the last one is like if you want to have the signal to know what comes from the brain and to understand where the signal has been produced, you need to do this inverse problem, which is not a very easy task. And I will go a bit more into details. So here maybe we can spend some time on this schematic. So you have this would be the electric field created by the brain, and you see that when you create an electric field, you have the magnetic field around it, and then you can measure it like through what we call magnetometers. And so you use two types, like gradiometer, which gives you the gradient of the field, and magnetometers, which gives you something more on the what is the magnetic field. And basically what is important to know also is that so Igor is saying that it's like 1 kilohertz, so millisecond. So yeah, what is important to know is that what we measure with images is like it's not one neuron, it's like thousands of neurons, which need to be oriented in more or less the same direction, so that these currents sum up, and then you are able to measure something which is big enough. Because if you magnetic field of one neuron it's not possible to measure it, at least outside the brain. Okay, so if you have no question, I will go a bit more on the how we try to know from where the signal is created. So these are just Boltzmann equations. So we know how this what are these fields, so electrical and magnetic, and we can use this equation to create a model and try to fit the parameter of the model to our data to see to try to see from where the signal comes from. So the model basically is just what you see here. It's like you can see these are some charges that move, and you will model it as some dipoles that create some currents, and at the end what you measure is this Y, which is basically just the sum of these currents, and of course you have to add some noise, which is theta here. And then, so this is your model, and then you try to fit the parameters to compare to your data, and try to see if you if you can understand from where the signal come from. Okay, so in EEG, what you measure is like the difference of potential between two electrodes, and in EEG you don't need difference. It's just like the magnetic field that go through your magnetometers and granulometers. Okay, so basically you map the brain with many dipole, like this one is like one dipole, which tells you in which direction the activity is created, and once you have this map, you try to reconstruct the data that you measure. Okay, so now once I will go quickly through the different step of processing the MEG signal. Existence. So to answer the question, the signal is basically the neurons communicate through electric currents, like generated by the move of ions. So that's what generates the signal. And then when you have many neurons in the same, for instance, the brain can be modeled as many columns, and when you have many neurons aligned in this column, you can sum up the reason the signal exists. It's mainly chemicals that moves. I don't know if it's what is not clear in this.
Can I talk to you?
Yeah.
Because I don't know what you measure. Must be some reason why you measure actually causing the signal. It's not a chemical reaction. It's not that something exists. Something must be a reason the signal exists, and it's very different from normal behavior of the organism, right? So what you really measure?
What we measure is the electrical field produced
by
the neuron. So it's like a cable. A neuron is a cable that sends information through this cable, so you can.
If you're standing still, and I measure your movement, you will not be moving. But if I push you, you start moving, and this will be a reason that I push you. And this is what I'm asking: what causing the signal?
Your brain is constantly moving.
Yeah, but you have to know what you measure. Why? It has to be synchronized to something.
No, your thought as, your brain is
all right.
always...
I listen more. Okay.
Okay. You want to?
No, I just wanted to add that basically the way in which the brain processes information is in this complex interaction between neurons, that it's neurons are interacting between these electrical signals. So basically all the processes that your brain is controlling, your respiratory, your thoughts, everything is controlled by these signals. So the brain is always working, and you're measuring some projection of those electrical signals.
I understand this one, but I also understand that you're trying to achieve something, but measuring response to something. And this is what I'm asking, what you're really focusing on. What is the excitation?
So there are many protocols. This I will not talk a lot about this, because the data that I use is called resting state. So we measure the brain of someone just sitting in this device that I just showed you.
You're measuring noise, basically.
Your brain is not.
It's always operating. Whatever you connect to the system, it's always some signal, right? And you can measure, and on the average, you will have some kind of random excitation or a random noise, really going nowhere. But if you want to make a reaction to something, you have to know what you measure with respect to something, right?
Yeah, sure. We cannot know what people are thinking about, for instance. So that's a matter, but of course, when you do a task, it's more precise. Yeah.
Okay, continue.
So there are protocols that do a task, and you can also look at resting state has been used a lot to know like in which what are the different states of your brain when just you don't do anything. It's also something that can be interesting. But of course, it's more precise when you have like a task and you do it many times, so then you can average and, as you said, like have a concrete signal that you know what it corresponds to.
But you don't even know if it's brain activity. It can be caused by something else, right? It's just a signal which is existing and you measure. You just have electrodes on the head and then you just measure something.
Yeah. Well, I mean, when you measure, I'm not sure I understand what you want to say, but yeah. I mean,
All right, let's say we look for, I'll come back to it.
Yeah. So when you have this signal, of course, it's very noisy, as I told you, because there are many patterns that are present in these signals, and the most easiest one to remove are like the blinks when you blink, when you have some eye movements, and these are example of also like the breathing, and you also have like your cardiac properties that you can remove. So you try, when you what is called pre-processing is you try to remove all the artifacts that you can, and to try to have to hopefully have something that is as close as possible to what only the brain created. So and then we, as I told you a bit before, you compute a forward model. So you create your source model, and you say, okay, this is, yeah, you create your model and you try to compare it to the measures, and then this is what you obtain at the end. You have like some dipoles that you expect to have been created, the signal that you measure, and you have them spatially distributed. Yeah, so yeah, this is just to show you, like, this is an example of a dipole. So it has a direction and it has given charge, and we know that given this, it creates a certain electrical field and some magnetic field. And so yeah, this is just equations that are used to find this inverse problem and try to minimize the error between the signals that you create with your model and the signal that you measure. And then you can analyze it in many ways. So the two big categories are connectivity analysis. So you can use different techniques like correlation, classic correlation, like Pearson, and you can also use spectral correlation or directed connectivity. And then there is the other part is also the frequency analysis, where you can look at power spectrum, for instance, and you can also look at the link between time and frequency, and also use like signal processing, like waveplates. Okay, so with this kind of analysis, what we know is that you have some changes in the oscillation that we observe in different parts of the brain. So for instance, what is called theta, so the different oscillation possible have been partitioned in different what we call bands of frequency, and theta is like around from 4 to 8 hertz, and so we see that theta is increasing in some central part of the channels, and beta is like from 13 to 30 hertz, and this one is decreasing. So this is this kind of data of results that we can have with this kind of analysis, and this is what it looks like if you look at the projection in this is on the channel side, so not on the source side. And then you can use more fancy methods. So instead of looking at the total power, so the total power associated to some bands, you can look at something more time results. So you can look at what is called beta burst, for instance. And it has been shown that there are some changes. So these are changes. What I show you is like changes between PD patient and healthy controls. Okay, but this is still something quite complex to interpret on the disease level, and we cannot. Yeah, we can say beta is increasing, beta burst is maybe decreasing, but then we need to do some model to understand the things, and one intermediate. So okay, there is another thing. Sorry, I forgot that. We can also look at how the all frequency. So in this case, it's like frequency peaks. You can look at how it changes between PD patient and healthy control, and this is from our study. What you can show is that when you look at the peak frequency, there is what is called a spectral slowing, so a shift in the distribution of the peak frequency towards the low frequency when you have Parkinson's disease. So Parkinson is in blue, and on and off is on medication, which is just dopamine that we give to the patient. And what you see is that when we look at the difference in the different brain regions of these oscillations, they are not significant. So it's kind of hard to interpret this slowing because it seems that there is some regions that shift, but that decrease, and some other increase. So it's kind of it's still hard to interpret this spectral analysis. So somehow we have to go beyond this classical spectral analysis to get more insights. And what I will explain in the end of the presentation is what we did is to look at something that is kind of oscillation are in fact on top of something else. So I will explain you. So what we measure, as I told you before, is like, so these are like spikes representation. So it's like electrical pulse that the neurons send to between them. And what we measure is like the summation of the synaptic current. So when a neuron sends a pulse, it will increase a bit what we call the membrane potential, which is a kind of electrical state of the neuron. So it will increase this potential, and it will have a different shape depending on if this information is inhibiting or excitating. So in the inhibiting case, you see that the time scale is very long compared to the excitatory case. So when you sum up this signal, you end up with what you can measure with, so LFP is the electrode in the brain, or EEG or MEG. It's like this sum of signal, and we see that inhibitory will have like slow, lower frequency components than excitatory ones. And when you look at the power, so how the frequency, different frequency are distributed, you see that you have much steeper power spectrum for inhibitory neurons. So this means that we can kind of have an idea on how this excitation and inhibition is activated or takes place in the brain just by measuring this MEG signal and looking at what we call the power spectrum. And in fact, when you look at, so if you look here, the curve is in fact what we measure when we measure this signal it's what we call it follows what we call a power law. So you have this kind of 1/f type of curve when you look at this power spectrum, and on top of it, this has a bump representing the oscillation present in the signal. So this steepness, in fact, can be rather quite well represented by this 1/f curve. So that's what we will use as analysis to try to infer the excitation and inhibition changes over the cortex. Okay, so this is to tell you that we, from this question and how we expect a change in excitation and inhibition, and now maybe we have a measure, a way to have at least an idea. It's not perfect measure, direct measure of excitation and inhibition, but it's like a good way to know how it changes. So now I will explain you a bit what our data and how we computed this 1/f, what we call 1/f exponents. So our data is like two different groups, so PD patient and healthy controls. We use, as we talked before, resting state, so people are fixing across, and we measure their brain activity for 8 minutes. And we have, then it's like 1 kilohertz, but then we downsample because to go faster in the analysis. And then so we have so we have 2 sessions. Like people are first deprivated from their medication, so they stop medication for at least 12 hours, and we have a first measurement, and then they take their medication, and we measure again. So yeah, these are details. So we use some low pass filter, and we use this source reconstruction. So we try to know from where in the brain the signal is produced, is coming. And we have at the end like 44 sources, and these are the details. So we use a given atlas which partitions the brain in different regions. Okay, so what we end up with is a signal like this. We have this for each brain region and for each patient. And then what we use is we use 50-second windows, and we then increment this window in order to compute this power spectrum for each of this window, and have also a temporal idea of how it evolves. So you compute this power spectrum for each window, and then you increment. So you end up with what is called a spectrogram. So here is this 1/f. It would be like for one given time, and then you just concatenate the results. And then for each of this power spectrum, we fit this 1/f using an algorithm which is called FOOF, and so basically it finds the best 1/f, and it models the bumps on top of this 1/f curve with Gaussian. Okay, so this gives us some parameters, kappa and lambda. So what we are interested in is this lambda, which will tell us how steep is our power spectrum. So it will give us some insight on how excitation inhibition changes. So in that case, how it changes over time. So this is like an example of a trace of lambda. It will be usually close to 1. And then we will look at this lambda in the different brain regions and different patients. So we will denote, this is just notation. So lambda bar will be the average over time of lambda for a given group, and then we'll average over different groups with this notation, mean and STD. Okay, so what we end up with is something like this for healthy control. We have inside the brain region is like the mean value of lambda, and on the border is how it the standard deviation of it over the different patients. So you see that the blue regions with the blue borders, there is a small variation between patient, and the higher, so the purplish region are the one with more variation in between the group. So this is for healthy control, and if you look at PD patient, you see that globally there is an increase of this lambda, so of this exponent over most brain region. You also see that you also have some more variability, for instance, in the posterior part here. Okay, so to have a better visualization of this, we just take the gravity, so the center of mass of the different brain region, and we project these results on the brain. And it looks like that at the end. So this is the same as previously, but projected on 2D brain, whole brain there. And what we see is that we have this kind of gradient that looks perturbed in Parkinson's disease, and you have this increase. And you can plot the difference between the two groups, and you see that there is not a big difference between on and off medication, but as soon as we compare to healthy control, some brain region appears to be significantly different. So this lambda is significantly more than in healthy groups. Okay, so and this significance is mainly present in sensory areas. So yeah, this is just to summarize. So this lambda is associated to EI ratio, so we have some disturbance of the gradient, and an increase of lambda means increase of inhibition or decrease of excitation, because we look at the ratio what we obtain, or both of them. And we also see that even though healthy controls, even though it's smaller in healthy control, it's also the variance is like the border. This is the variance over the groups. So it's the variance is even though lambda is smaller in healthy control, it fluctuates more in this group, which is kind of counterintuitive. Okay, then maybe I will go a bit faster. So what we looked at here is like how this correlate with UPDRS, which are like tests, motor tests of a patient. And what we can see is that this lambda does not correlate so much with the results of the tests. So it seems to correlate, but not significantly. But if you look at the correlation with age, there we see quite significant correlation. Maybe meaning that Parkinson is like kind of accelerating aging, because what we see is that this correlation is very present in significant in this sensory area. So we can and we don't see any correlation in healthy people. So maybe Parkinson is like kind of accelerating aging, but because we know that when we get older, the first things that are impaired are mainly the sensory areas. So this is maybe an assumption to study further. Okay, so just to conclude on this part, we see that EI ratio seems to be correlated with age, but not with UPDRS, and we have a decrease in this is wrong. We have a decrease with UPDRS and an increase with age. Okay, so this is very nice, but still we know we have maybe some insight on how excitation inhibition changes, but we still don't know really how to interpret it and how to maybe the final point would be to help curing the disease or treating the disease. So I think that a good way to do this is to use modeling, and we know that excitation and inhibition is very important in modeling. So at least in neuroscience. And so the idea now is to look at a network like this and to model each brain region as also a network. So it will be a network of networks. And we want to change the properties of the different networks. So try to change so yeah, this is model, and then we will change the ER ratio in different way and try to see how it impacts different nodes dynamics. And it can modify it in many ways. So you can it can create, as I told you at the beginning, in the basal ganglia, it can create some spike, changing the spike frequency, changing the synchronization, oscillation, etc. So all of this is kind of suitable to study with some models. And then, yeah, of course, the endpoint would be like to try to know how changing ER ratio can modify and then maybe lead to some disease, because we know that excitation inhibition ratio is not altered only in Parkinson disease, it's altered in many other disease, brain disease. Okay, with that, I will thank my different collaborators, especially Arvind, who hired me, and really enjoying what I'm doing thanks to him. So yeah, with this, I will leave you with these 2 videos, and I hope you can guess who is the PD patient and who is healthy control. Maybe I don't know if you have any question. I would be very happy to answer.
I will stop the recording now.
Yeah.